on that day, right? His work was not published. Secondly, his concept of applications of biological applications of biological applications. Okay, so he applied mathematical expressions upon the biological theories, which was completely new and as an explanation for the apparently continuous variation seen in the Mendel's work. Thirdly, Mendel approach using mathematics formulas to explain the biological phenomena in the genetics was totally new. As you know that factors, the word factors was given by Mendel, but as per Mendel, characters are controlled by, let us take any one character which is under the control of factors, These factors later on coined as a genes by the scientist called, by a great scientist called Bateson. So these genes, whatever they are, okay. In 1900, three scientists came. Hugo de Vries. Who is that? Hugo de Vries. Hugo de Vries was familiar with the variations in Oinothera lamarckia, isn't it? And Correns, if you take the Correns, color Correns, Setchmark, and independently rediscovered Mendel results on the inheritance of characters. Okay. Also by this time, okay, and Hugo de Vries was a scientist who noticed mutations for the first time in Oinothera lamarckia. These three scientists individually worked on the Mendelian principles and republished them. Scientist was able to carefully observation on the cell division. They put their observations on the cell division. This led to the discovery of structures in the nucleus. The nucleus contains certain kind of double and dividing structures, and these structures were observed during the time of cell division. Okay, these were called as a thread-like colored structures called chromosomes. What are they called? Colored bodies called chromosomes. So why? Because they are visualized under microscope by the staining process. By 1902, chromosomes movement during meiosis had been worked out. So after discovering the chromosomes, two major scientists came to the field, Walter Sutton and Boveri. Theodore Boveri noted that the behavior of chromosomes for the first time. These chromosomes and their behavior was parallel to the chromosomes behavior of the certain kind of structures called genes. Okay, these genes and their behavior generally parallel to the chromosomes even. So chromosomes movement to explain Mendel laws during the time of inheritance. Recall that we have studied the behavior of chromosomes during mitosis cell division. Mitosis contains four different types of phases: prophase, metaphase, anaphase, telophase. Okay, and during that phases, you can also observe the chromosomal development. During prophase, chromosomes are just like a small mass, okay, and they are thread-like structures which are in the form of small mass. But whereas during meiosis, you can see the process of reduction division. What is that? Reduction division. The important things to remember are that chromosomes as well as genes occurs in pairs. Chromosomes and genes occurs in pairs. That is very most important point. In fact, the two alleles of a gene pair, if you take the two alleles Let us take the two alleles of a gene pair. Two alleles of a gene pair, if you carefully see, are located on the homologous chromosomes. They are located on the homologous chromosomes at the specific site, at the specific location. You can see the possibility 1 here, possibility 2. In possibility 1, one long and orange and short green chromosomes and one long and short red chromosomes at the same pole. Let us take in the meiosis 1 anaphase. Here during meiosis 1, see the behavior of the chromosomes, these colored bodies, yellow and red colored bodies, okay, orange and green colored bodies. During meiosis 2 anaphase, they will be separated. They will be separated. You can see the separation, right? So orange chromosomes and short green chromosomes, yellow bodied chromosomes and red colored short chromosomes. But when they enters into the germ cells, okay, or mother cells of the gametes, germ cells, individually they separated. Individually they separated. You can see that. So they individually separated, enters into the individual cells. In the meiosis 1 anaphase also, you can see orange colored chromosomes and short red colored chromosomes, yellow bodied chromosomes and short green colored chromosomes. Okay. Finally, when the germ cells are formed, they segregated, okay, they separated, which explains independent assortment. What is that? Independent assortment. Independent assortment of chromosomes in the germ cells during meiosis. This explains separation of the genetic material, separation and assortment of genetic material in the germ cells. Finally, Sutton and Boveri argued that pairing and separation of chromosomes would lead to segregation, of a pair of factors. Segregation of pair of factors they carry. Finally, if you take the Sutton, united the knowledge of chromosomal segregation with Mendelian principles and called it as a chromosomal theory of inheritance. Okay, this experimental verification of the chromosomal theory of inheritance, okay, by another scientist, Thomas Hunt Morgan. Who is that? Thomas Hunt Morgan and his colleague led to the discovery of basis of variation during the reproductive phase or reproductive process, sexual reproductive process. Morgan worked with a tiny fruit fly called Drosophila. What is that? Drosophila melanogaster. What is that? Drosophila melanogaster, which is a common fruit fly, one of the fruit fly. Now, it is suitable for his study. They could be grown on a simple synthetic medium in the laboratory. That's why he has selected them. They complete their life cycle. Their life cycle will be within 2 weeks. And a single mating or single reproduction, if you take the single mating could produce a large number of, what are they? Single mating can produce large number of progeny files. That's why he has selected them. Also, there was a clear differentiation of the male and the female bodies. So that's why he has selected this. So that's all for this chromosomal theory of inheritance. These points we have to note down and remember. That's why I am giving the notes on the side of the slide. Thank you. Don't forget to like. Have a nice day.